Hi class. So today we're going to cover motor proteins, and this lecture really goes really closely with last lecture. So sort of take them as a group, and you'll see how they come together. Because, in fact, the motor proteins actually move on the cytoskeleton. And so, if you remember last time, we learned about the cytoskeleton, specifically actin filaments, microtubules, and the intermediate filaments. And we saw how dynamic they were. And even though those individual filaments are dynamic, that alone doesn't bestow the cell with all of its dynamic properties. It actually has motor proteins that move along the cytoskeleton and give it that dynamic character. And there's three major motor protein groups, the myosins, the kinesins, and the dyneins. And the first part of today's lecture is going to be looking at these motor proteins specifically, and then seeing how they function up until like muscle cells. And then in the sort of second part of today's lecture, we're going to be looking at sort of more wide cellular processes where these motor proteins in the cytoskeleton sort of come together to accomplish various things in a cell, like basically mitosis, as well as the movement of cells and even connections and setting up synaptic connections and whatnot. Okay, so let's look at the first motor protein that we're going to talk about, and that's myosin 2. So myosins as a family, they were the first major motor proteins to be discovered. And, you know, part of it is that myosin 2 is actually enriched in muscle cells, and it's needed for contraction. Okay, and we'll see a lot about this. And just please know that myosin 2 is the motor protein in muscle cells. So here's just a schematic of myosin 2. It has a coiled coil of 2 alpha helices, and then there's actually these light chain proteins down here near the sort of associated with the neck region, and then there's the N-terminal head region. And so it has 2 heavy chains and these 4 light chains, and these are assembled via the coil and eventually can get assembled into large bundles, as we'll see in a second. So here's just an electron micrograph of myosin 2. You can sort of see it over here. Here's 4 separate images, and you can see the coiled coil, and then you can also see the head group. And the light chains right here are also buried sort of in this neck region. Okay, so it's very recognizable structure on an EM. And in fact, the myosin 2 thick filament, which is basically how it's assembled in a muscle cell, this thick filament here, it's basically a bipolar filament. And here's an electron micrograph of it above. And all of the various myosins are actually sort of attached together and bundled. And so they form this sort of bipolar filament with the head groups. And it turns out that because it's bipolar, it sort of goes in 2 directions, right? And we'll see in a second how the myosin head groups sort of move on actin, but they hydrolyze ATP and they always walk towards the plus end. So myosins are plus end directed actin motors, okay? If we want to take a little closer look at it, what you'll see here is you have the myosin tail, the coiled coil, and it's sort of assembled in this thick filament bundle, and here you have the actual head groups shown here, and they sort of stick out, and that's what attaches to the actin in a muscle cell. Okay, and so the myosins actually have motor activity. Okay, so if you were to take a slide and you were to coat myosin on it, it has this head group, and the tail is actually going to be sort of associated with the glass slide. And if you just put actin filaments on it, they're going to have direction, and they're going to sort of move, provided that you're supplying a ready supply of ATP to the glass cover slip. And so as you look at this fluorescence image above, where you have these 2 actin filaments, one's sort of colored in red using rhodamine, and the other one is colored green here using a fluorescent, green fluorescent tag, and you can see that they move as you sort of look at this time-lapse video here. So the green is moving downward, and the red one is actually moving upward. Now it turns out that myosins are actually a whole superfamily of proteins, okay, and they're very similar. They all have a sort of a motor domain, and then they have various tail domains. You don't necessarily have to memorize all these. A couple of the ones that I do want you to know, though, and how they differ is myosin 5, which we'll see in a little bit. It tends to move little vesicles around a cell, and also myosin 2, which is composed of the myosin thick filament in muscle cells, and also it's involved in lamellipodia movement, as we'll see in the second part of today's thing. And so make sure you know the difference between them and refer to them as myosin 2 or myosin 5, depending on, when you're talking with someone or when you're answering a question or something like that. But it is important to note that the motor domain here, the head domain, is highly conserved, and then they have sort of variable tails. And here's just some schematics of them. Here's the myosin 2, the neck region, and where you'll have the light chains, and then here is myosin 5, which also has a coiled coil at the end. And just remember, these are all plus end directed, so they will walk towards the plus end on an actin filament. The other family members are the kinesins, and these are microtubule specific motors. And if you guys remember back to the first day of class when we saw that movie of the animation and we saw the kinesin sort of moving along a microtubule, you can think back like that. And some similarities between these and myosins, they all have a highly conserved motor domain. And it turns out humans actually have 45 different kinesins. And most of the kinesins are actually plus end directed, but there are a few rare exceptions. Kinesin 13 and 14 family members, they're actually minus directed. And these kinesins were actually first identified in the squid giant axon. Okay, so you can actually take the major axon out of a squid, and you can purify the kinesins out of it and actually isolate them in vitro and show that they have motor activity and can move on microtubules. Here's just an EM of kinesin. It's a little harder to see in this image than the myosins were, but you can see sort of the head domains, and then you have the coiled coil. The third family of proteins are dyneins, okay? And there's a couple of different kinds of dyneins. There's cytoplasmic dynein, which is shown here, and you also have ciliary dynein. And these guys are actually minus end directed microtubule motor proteins, so they'll always move to the minus end, whereas in certain instances the kinesins can either go towards the plus end, or there's occasionally minus end directed ones, and these move on microtubules. So once again, actin or myosins move on actin, kinesins and dynein move on microtubules. Now it turns out that they both use, myosin and kinesins also use ATP, and they hydrolyze ATP to get some of their movement. And we'll look at the power strokes of these two motors in just a second. But what you guys just need to sort of appreciate here is there's some structural conservation between these two motor proteins. And here's the ATP binding domain in myosin and in kinesin, and I'll leave it up to you guys to sort of look this over and see some of the structural similarities. You can see ATP is bound in both things. So there's very little amino acid identity between the motor proteins, but structurally they're very similar. And this goes back to lecture 3, where I was sort of telling you guys about how certain domains are used over and over again, and this is just an example where the ATP binding domain is used in both motor proteins, even though there's very little amino acid identity between the two of them. Okay, so now let's get into the actual mechanism of movement of these motor proteins. So we're going to start off with looking at myosin moving on an actin filament. So you can see this actin filament sort of shown here in sort of a peach color. And there's different stages that the myosin goes through, and it's depending on the state of ATP present. Okay, and these sort of states are basically attached, released, cocked, and then force generating, and then it goes back to attached. Okay, so let's look at them, and I encourage you guys to read this closely because it's got some very good information in it. But in general, what you have is attached, okay, and this is sort of the state where it's bound to an actin filament after it's been fully undergone its power stroke. There's no ATP present, and it's what's sort of called locked in a rigor state. Okay, so it's bound here and it's locked in a rigor state. And this is actually happens with rigor mortis, right? This is how it got its name. And so then when you add ATP in, the myosin thick filament actually detaches and when it binds ATP. And then it hydrolyzes that ATP, and this is called the release state when it's just bound with ATP. And then it hydrolyzes the ATP to ADP and inorganic phosphate. And that causes it to sort of shift forward a little bit. You can see the dotted lines here where it used to be, and then once it's hydrolyzed ATP to ADP and inorganic phosphate, it's become sort of cocked. Okay, and then upon release of the inorganic phosphate, it reattaches to the myosin, and then when the ADP is actually released, that's when it undergoes sort of the force generating power stroke movement, and then it returns to its attached / rigor state. Okay, so that's a pretty straightforward thing. You guys should really know how the power stroke works here for actin. This is something I really want you guys to appreciate, right? And this is just one myosin head on the myosin thick filament. So this is actually what's happening during a muscle contraction. These four sort of events where you have attached, release, cocked, and then force generating. And it all has to do with the binding and hydrolysis and release of ATP. Okay? In the case with kinesin, and once again, I encourage you guys to go back and look at that video we saw on the first day where you saw the kinesin motor sort of walking along a microtubule, pulling that giant vesicle along. And so in the case with kinesins, unlike myosins, they travel in sort of a stepwise fashion, right, where they sort of lumbering along, and that's how you saw it in the video, right? And they take like 8 nanometer steps, and the coiled coils play a role in the directionality, as we'll see in a second. But in general, what you have is your lagging strand will bind ATP. That'll basically hydrolyze the ATP to ADP and inorganic phosphate, causing it to sort of swing forward. And then you'll have the release of the inorganic phosphate and an exchange of ADP for ADP on the leading sort of foot here, the kinesin head. And then what ends up happening is it swings around. Okay, so you'll have ATP now on the trailing, and ADP, the ADP bound form sort of swings forward, and then the whole process repeats again. Okay, so this is sort of in comparison to the power stroke of the myosin, and you have this sort of kinesin walking where it sort of uses the hydrolysis and exchange of ADP to ADP to sort of walk along the microtubule. And the directionality of the comes into play with the coiled coils. And here's sort of how that works. So you have kinesin 1, which is a plus end directed motor, so it's going to move to the plus end of the microtubules, and here you can see a sort of a space filling molecule of the microtubules, and you can see that the kinesin head group is sort of associated with it, and then the unbound head will sort of move along towards the plus direction by, hydrolyzing ATP and exchanging the ADP for ATP. Okay, so that Kinesin 1 is a plus end directed, and one of the things you should note is that the head in this one is actually on the N-terminal region. Now for the minus end directed motors, they tend to have the head group in the C-terminal domain, and this is Kinesin 13, and it sort of just walks in the opposite direction towards the minus direction in the same sort of mechanism that I talked about right here. The third class of molecules, motor proteins, is dynein. Okay, and dynein is different from kinesin and myosin, both structurally and in the way that it works. Okay, it sort of forms this ring-like gearbox structure. Okay, and it uses the hydrolysis of ATP to almost work like a ratchet. Right, it has this AAA domain, and then it has a stalk domain, and they can actually attach to the microtubules, and they basically use the hydrolysis of ATP to sort of move, and it just sort of ratchets along. And here's just some electron micrographs of the dynein molecule. Now, as you guys can probably imagine, with the 45 different kinesins and the whole, the 15 or so myosins, these have sort of been adapted for different functions. Okay, and in the case with myosin 2, we've seen that it forms that the myosin thick bundle that's used for muscle contraction, and it has actually a sort of a shorter lever arm. Okay, so it doesn't go as far, whereas myosin 5 has a much larger lever arm, and myosin 5 tends to move small vesicles around. Some characteristics of the motors that I think you guys should know. So myosin is the fastest, is faster than kinesin, okay, by quite a bit. And so the myosin moves faster than kinesin, and we saw that sort of in the video. If you sort of think about it, they had the myosin just sort of, or the kinesin sort of just lumbering along as it moved down the microtubule. And in general, myosin is rarely bound. It's only bound 5% of the time, whereas kinesin, at least one sort of foot of the molecule or the motor domain is almost always bound to the microtubules. And as we saw in the video, the motor proteins actually move organelles around. Okay, and so here's just a beautiful cell, and the microtubules are stained in red, and then you have the Golgi apparatus, which was a large vesicular complex that comes after the ER, and we talk about the secretory protein. We'll learn a lot more about the Golgi. But here you can just see the Golgi arranged, and it's sort of attached to the microtubules. When you depolymerize the microtubules with nicotazole, which actually causes depolymerization of the microtubules, the Golgi actually falls apart and breaks into small vesicles. Okay, and we'll see when we cover the secretory pathway that the kinesins are actually tethered to the Golgi and to the ER and to what are called vesicular tubular clusters, and they actually drag ER membrane blobs to the plus end. Okay, and the cytoplasmic dynein is another motor protein that actually is attached to vesicles, and this walks them towards the minus end. Okay, so kinesin in general are going towards the plus end, and the cytoplasmic dyneins are going to the minus end. With the exception of kinesin 13 and 14, they'll move to the minus end. And here's how dynein moves vesicles. So it's actually attached to this large complex called the dynactin complex. So here is your vesicle cargo with your membrane glycoproteins associated with it. Then you have your dynein attached to the microtubules. And then there's this dynactin complex, which consists of spectrin, anchorin, and the sort of the actin-related protein 1, or ARP, which we saw before was actually involved in we saw that ARP2 and 3 were involved in nucleation of the actin filaments. So here's just another actin-related protein that's attached to it. And so it's, I think, widely believed that the ARP1 protein binds to the spectrin here to help assemble this thing. And this can move vesicles down the microtubule in the minus direction. Okay. Now, a great system for understanding the movement of vesicles on basically myosin and actin, or on actin and microtubules, are melanocytes. Okay, and these are just cells that contain pigments in different organisms. Fish actually contain them, and then sometimes you can, if you guys have ever had fish, you know, depending on the condition of the fish, he will actually change his colors if he's scared, or you know, or if he's she you know, can change their colors depending on certain aspects. Okay, and so if you just look here at this image, these sort of little black circles here are actually pigment-containing vesicles, and they can actually move within the cell. And it turns out that these pigment vesicles are often bound by myosin 5, which is attached to the vesicles in melanocytes, and will move these vesicles on actin in the plus direction. Okay, melanocytes also can move on microtubules with the Dynein and with the kinesins. And here's sort of a example of how that's all regulated with the release of cyclic, or with changes in cyclic AMP levels. So here it's just sort of dispersed, and you have these vesicles attached to the microtubules. And then if you basically decrease cyclic AMP, it will cause them to walk to the sort of minus end and the centromere. And then, you know, if you increase cyclic AMP, they'll actually move out to the plus end. Okay, so sometimes you'll have plus end directed motors moving them outward, or minus end directed motors moving them inward, and you have the changes in cyclic AMP that help regulate it. And here's just two images where it's these pigment vesicles are either dispersed or they're sort of aggregated in the center. Okay. And here's sort of just a great image of the movement of these pigments, and both Dynein and Kinesin are associated with these pigment vesicles, or the pigment granules. And you can see here you'll have basically the plus end directed here and the minus end, and so you can see that this pigment vesicle is actually moving to the minus end, which would mean it's likely being moved by Dynein, and then it can actually move back out using the kinesins to the plus end. Now, motor proteins are not only needed to move vesicles around, they actually move cellular molecules. Okay, and a great example of this is movement of messenger RNA within a cell. Okay, so here we have our favorite little Saccharomyces cerevisiae, our little yeast cell, and it's actually budding. This is the bud cell, and here is the mother cell. And it turns out that ASH1 is a messenger RNA, and that gets localized to the bud tip in Saccharomyces cerevisiae. And one of the things that it needs for this is the actin-associated protein formin, which actually polymerizes the actin on the plus end, and that helps move the messenger RNA to the tip of the bud site. And here's basically how that works. You have your actin filament oriented, so the plus end is near the edge of the cell. You've got formin, which is involved in basically the polymerization of the actin at the plus end. And then you have myosin 5 here, which is associated with the She3 protein. And then you have your ASH1 messenger RNA, which is bound by the She2 protein, and the She2 and She3 protein come together, and the myosin is bound to myosin 5 is then can walk along the actin filament towards the plus end, and that's how the messenger RNA gets localized to the bud tip here. Okay, so you'll see when we sort of cover nerve cells that neurons actually use this same process, and they can localize messenger RNA close to the synapse so they can readily make neurotransmitter if needed. Okay, so in the past, I really haven't held students to know this, but this year I think I'm going to have you guys, really sort of understand this process, because messenger RNA transport in the cell for selective translation is actually a pretty important concept, and we sort of saw that already when we were looking at Drosophila, the embryos, and when we were looking sort of at the different stripes in the Drosophila embryo. And so just please take an effort to really understand how this works. So, if a question comes up, you guys will be able to answer it. Okay, so now we're going to basically look at assembly of the myosin thick filaments that occur in muscle cells, and then we're going to look at how muscle cells function. Okay, and this is another area, over the next 5 or 6 slides, I really want you guys to sort of understand this along with the mechanisms of how the myosin works for the muscle contraction. Because I really think if you can explain this and you can understand it, then you really truly get a good feeling for, how muscles function. And this is a huge problem right now with an aging population. Because there's this disease called sarcopenia, which is basically sort of muscle wasting, where elderly people tend to lose muscle mass, and that inhibits their mobility, and it causes a large increase in healthcare costs when people can't be alone and independent. So understanding all of this, I think, is really important from a human health perspective, and as well as an animal health perspective. So I want you guys to know this. Okay, so let's look at the assembly of the thick filaments, because this is where it sort of starts out. You have your myosin sort of in an inactive state, where you have your myosin 2, and then you have your myosin light chains. There's actually a kinase called myosin light chain kinase that will actually phosphorylate the light chains. And this causes spontaneous self-assembly of these bipolar filaments, where these myosin tail domains, the coiled coils, are coming together to make this bipolar filament. Okay, and so making this bipolar filament, it can technically walk to both plus ends of an actin filament. Okay, and so this is the first step in the assembly of that bipolar filament, where you have this thick filament assembled for muscle cells. And we already saw sort of a better image of the myosin thick filaments, but here it is again. And once again, the myosin light chain kinases will phosphorylate the myosin light chain and cause it to assemble these thick filaments. Okay, and this is really where the power comes in a muscle contraction. These two bipolar filaments sort of moving in opposite directions, pulling the actin together in the sarcomere. And actually, here's basically what a muscle cell looks like. You have these things called myofibrils that are in the muscle cell, and it turns out that muscle cells are actually pretty large cells, and they basically form by fusion of many monocytes. And it turns out, because of that, they're actually multinucleate. Okay, so a single giant muscle cell will have many nuclei, and then it will have these myofibrils actually contained in them. And each one of these is then broken down into an individual sarcomere, these little sort of discs assembled end on end, and these myofibrils are actually called sarcomeres. So here's the staining of a muscle cell, and once again, the myofibrils are stained in red here. They're pretty easy to see, and the nuclei are stained in blue. If you take a look at the myofibrils in an EM, here's what they look like. You basically have this repetitive array here of these individual sarcomeres. And so a sarcomere is basically from one Z disc to another Z disc. Okay, that's one sarcomere. And now in these sarcomeres, what you have is a dark band, and that's the myosin thick filament contained in there. So if you go back a couple slides to that EM, these are now all assembled. And then you have sort of the light band here, which is basically containing all of the sort of actin filaments of the perfect length. Okay, and so if we look at it sort of closely, you have your dark band here, and it's got this M line, and that's the midline of the myosin thick filament, and then you have your Z disc on either end. And if you guys, not to spoil things, but if you guys remember to last lecture when we were talking about actin filaments, I said this protein CAP-Z actually binds to myosin to cap it, and so on the plus end, and so you'll see in a second that this Z disc actually contains the protein CAP-Z. Okay, and so when there's plenty of ATP around and high levels of calcium, you'll get a muscle contraction where the myosin with the myosin thick band and its bipolar orientation, actually the motors attached to it and actually pull the two Z bands together, and that leads to muscle contraction. Okay, and we'll see that closer in a second here. But here's basically an EM of an insect flight, sort of arranged in a similar sort of fashion, where you have all of these sort of filaments here and motor proteins in it that basically form these sort of fibrils that can be pulled together and used for insects to fly. Okay, so here's just sort of a better description of the sarcomere. Okay, so once again, you have your myosin thick filament in the middle with the M line, and these are bipolar filaments, so they can basically walk to the plus end in both instances, and these the actin filaments, which are sort of shown here in green, or sort of they're actually not colored because you have nebulin and some other things bound to it, but you can sort of see the clear little white circles there. That's the actual actin. And the plus end of the actin filament is bound by CAP-Z, which is sort of shown here as being also associated with the Z disc. And then the minus end is capped by tropomodulin. Here on the minus end, and then you have the myosin thick filament attached to this molecule called titin, and the actin filaments are actually sort of held perfect distance apart. Okay, and so when there's plenty of ATP in here, the myosin thick filaments will actually both move to the plus direction, which actually causes the Z discs to sort of come together, right? And that's a muscle contraction. Okay. And so, once again, just sort of understand the sarcomere and how it all functions, and we'll see the regulation coming up in the next slide. Okay, so the muscle contractions are actually regulated by calcium release, and we'll see how that sort of works. But calcium release actually starts when an action potential comes in contact with these transverse tubules, or T-tubules. Okay, and they basically are adjacent to the sarcoplasmic reticulum, which, like the endoplasmic reticulum, has large calcium stores in it. Okay, and so what ends up happening is you'll sort of, here's just a nice little image of the sarcomere here, and you've got the transverse tubule that goes around, and then you also have this sarcoplasmic reticulum that's sort of shown in this light bluish white color that's here. Okay, and so when an action potential travels down a neuron, and eventually it will cause a depolarization in the T-tubule membrane. Okay, and that has a voltage-gated calcium channel in it. Okay, and when the T-tubule gets depolarized, the channel actually opens, the calcium channel, the voltage-gated calcium channel actually opens, and calcium flows out. And then the release of calcium binds to a calcium-gated calcium channel on the sarcoplasmic reticulum. And that causes the calcium-gated calcium channel to open up, and then there's a release of calcium, which can then allow a muscle contraction. Okay, so basically what you have here is an amplified release of calcium. And the reason that you need so much calcium release for a muscle contraction is because you have these proteins called troponin and tropomyosin that are actually binding to the actin filament and preventing the myosin 2 motor protein from associating with it. Okay, so in low calcium, troponin and tropomyosin are associated with this actin filament so that the myosin 2 motor protein can't interact. And then upon calcium release, the troponin is released, and that actually releases tropomyosin, and it gives a place for the actin, for the head domain of the myosin motor to associate with the actin. Okay, and so getting back to this calcium release, that's actually releasing sort of inhibitors that are bound to the actin filament, and once they're removed, the myosin motor can then bind. Okay, and so here's just a sort of a way to look at it. Here's the tropomyosin blocking the actin binding site. This is actin right here. This is where the myosin head group would actually associate, and then when you add actin, there's a movement of the tropomyosin off of the actin filament so that the myosin 2 motor can associate and sort of pull the muscle together. Okay, so I hope that makes a lot of sense. Please know how a muscle contracts. This is just really important stuff that I want you guys to understand. And you can sort of see where all this would come into play. And here's just the heart, which is a major muscle in our body and a pretty darn important one at that. If it's not working right, you're likely going to have some severe health issues. But there's a mutation in the cardiac myosin which can cause an elongated or an enlarged atria. Okay, so, and that can be big problems. And so instead of myocytes like the muscle cells, the heart muscle actually has cardiomyocytes. And so, they basically function the same way to cause contraction of the heart, which then pumps blood to the rest of our body. So that's all I have for Part A. In Part B, we'll look at some more sort of combinations of motor proteins and the cytoskeletal filaments and look at how they sort of accomplish various functions. Okay, so with that, I'll pause the first part of the lecture.